how we got interested really in this topic was because we were interested in lung cancers in never smokers. So when you first think about lung cancer, you often think it's related to smoking, but there are at least 15% of cases actually that occur in never smokers. What's unique about lung cancers in never smokers is that they tend to be characterized by very few mutations compared with smoking lung cancer. So we were wondering what sort of risk factors could work on this context of very few mutations to drive cancers. We explored several risk factors, and we found that actually one of them was air pollution that was associated with lung cancer incidence, in particular EGFR mutant lung cancer incidence, which is common amongst never smokers. So this got us thinking a little bit more about how we could explore this. Perhaps we're thinking that air pollution, instead of inducing tons and tons of mutations, could actually work on pre-existing mutations. You might just have a single mutation in a nascent clone, but you might get a carcinogen like air pollution that comes by and creates this conducive environment for the cancers then to grow. So we started to explore this hypothesis. In our lab, we have some mouse models with pre-existing mutations in EGFR and KRAS. And when they were exposed to air pollution, we see that the number of tumors and size of tumors actually increases in a dose-dependent manner, suggesting that air pollution could indeed work in this mechanism. So that's in mouse. But in human, what we did was we also started to look at some of our normal lung tissue samples to see if we indeed have any of these pre-existing mutations. And we do see actually in about 16 to 19% of our normal lung tissue samples that these mutations in EGFR do exist, suggesting that they're present there for air pollution to act on. We also correlated the number of mutations that we see in normal tissue with age and see that there was a correlation, suggesting that you probably get these mutations due to an aging process rather than a carcinogen. And it's the carcinogen that comes later on then to actually drive the expansion of these little clones into a full cancer.